Hi everyone. So it's going to be two speakers. We're going to share the slides. How it's going to be smooth enough. So right. So we are the Tree of Life program, the Sanger. One of the main. Can I move? Yeah. Easier. Right. Thanks. So we want to make 6 scientific programs, and the goal is to make reference genome assemblies for every species on Earth. So we're starting with everything in the UK and then growing. We are many teams involved in that. We're about more than 60 people, and it's about making standard processes as well. So we've already done more than 1,200 assemblies, and those are currently new species for which we didn't have any assembly so far. And so far we're doubling year on year. So next year, by the end of next year, we'll have done another 1,000 or 2,000 more. No? Ah. Okay. And then building on what Arshad just said, we're following the methodologies, the standards provided by the NF Core tools. Because they're already there, they're very good standards. They make our things portable. Like you said, they can just run on the cloud. As long as you follow standards, it just works. So it's really great. And we've got all our documentation now laid out in a standard way on the pipeline website. That's a collection of close to a dozen pipelines. So here I've selected the main topics. Some of them have got posters out there if you want to talk to the authors. So it's about genome assembly, decontaminating the assembly, that's removing sequences from foreign organisms, helping the curation teams to make the best genome ready out of data, and then standard genome analysis for everything. So, Damon.
Hello. So I'm one of the 3 developers on TreeVal. We're currently running up to our 1.1 release called Ancient Aurora. And we sit between the decontamination of genome assemblies and the manual curation of those assemblies to effectively turn them from scaffold-based to chromosome-level assemblies. And to do this, we generate a lot of data, quite frankly. We make 46 different files, including their indexes, across a variety of formats in 13 major sub-workflows, 5 minor sub-workflows where they're essentially a sub-workflow nested inside another sub-workflow, and well over 100 modules at this point. For those of you that have seen the poster outside, I hope you can attest it's a pretty big pipeline. And we shovel all of this into JBrowse 2 that you can see up there. So each of those is a different data type that a curator can use to improve the contiguity of a genomic assembly. And these are some more. So we've got, it also generates high-C maps from Juicer for high glass and for pretext, which is a modifiable high-C map. So far, TreeVal has been built kind of halfway. Well, it started about a year and a bit ago now. Previously, we used something called GVal, which kind of died unceremoniously, in all honesty. But since then, we've been working on TreeVal, its successor for the Tree of Life. And we have done 70% of the assembly so far. Eventually, we will do everything. So that will be the 70,000 species that we estimate are in the UK and Ireland. And for each run, we use, for each species across the entire toll pipelines, we use about 8,000 to 10,000 CPU hours. For everything, that will be 630 million CPU hours over the next 10 years. This excludes the monsters like mistletoe, which took literal months to shovel through the pipeline. Obviously, this is a hell of a lot. So any sort of optimization we can do to bring this down will help out us, it will help out our infrastructure teams, and it will help out other users of the farm. And the issue is that we've come across is that Cicera platforms, their optimizer on there only does one pipeline run at a time. So, do you want to do this function? Yeah. Which I will come to more in a minute. The suspense.
The NF core modules we're using define our resources as low, medium, high, and have bins of 6 GB of memory each. Actual usage is usually not exactly a multiple of 6 GB, so you're bound to have a bit of waste there. But it's not just that. Actual usage is usually dependent on the input of your module or your software. There can be quantitative values like size, length, number of sequences, but also qualitative ones like just a clade name or things like that. And having unoptimized pipelines costs us a lot. If we underestimate resources, then the process will crash, and we have to be restarted, maybe multiple times. If you overestimate, and that's a lot of resources that we're paying for that actually we're not using. So we really care about using our HPC and the resources efficiently. That's why we're doing all these efforts.
So to combat this, we decided we needed to add some context to the execution logs. So what I've done is wrote a very simple, groovy module, which we shoved into the pipeline that collects some very basic statistics on the input of the pipeline. Obviously, by itself, this means practically nothing, but we have run this across 350 runs of the pipeline so far. And we push this through a summary stats script and generate some graphs for some actionable insights. Currently, we produce about 40 of these with plans for many, many more. But ultimately, I need to be able to drill this down to a per module, per process, add the max total resources against the peaks and requests. We also use the CO2 footprint module plugin that was mentioned earlier to kind of, we're DTOL, we're Darwin Tree of Life. The whole point of this project is to empower conservation. We can't really empower conservation if we're wasting resources by the bucketful. And both me and Matthieu essentially started optimizing these pipelines in our own ways and have kind of converged upon making a ton of graphs, which is quite nice. So this quite horrific looking graph, actually, this is an output of the script so far. So on the left, we have all the unique clade counts so far, the types of run, because TreeVal is run both in a full and rapid mode, depending on the data available, and the projects they belong to. So this is of one of the sub workflows in TreeVal that takes Hi-C data and maps it against the genome. The blue dots are it's the peak memory as a percentage against that requested. And the box plot is the average used memory as a percentage of that requested. So as you can see, that looks pretty horrific. We're using on average, maybe 30% of the memory we're actually requesting. So this slows everyone down. This means we can't get as much TreeVal runs done at the same time as we want to, makes other people on the farm wait for their pipelines to run. It's just not good all around. Some more examples. These are just some other examples of graphs that we generate. So Hi-C against duration, to try and see whether the total amount of Hi-C impacts it. It doesn't much. That gets pretty flat pretty quick. And that's just because it's parallelized. So pretty much every time Hi-C data is getting split into at least 6 chunks, run at the same time. It's pretty happy. We're also doing some categorization of these runs to see if there's any anything that we can drill down to. Oh, insects take X amount of resource, or as you can see, fungi are on average much less than the clades around them. This is an actual usable graph. So this is the amount of high C against the amount of memory, the peak memory for that that sub workflow. So using this, we'll be able to plug the trend line into the config, and we'll get it to start dynamically requesting X amount of memory per gig of high C data, which is quite cool. And obviously, we've categorized this down to the clade level, and there are some slight differences. Now, the ones that will make IT hate us. So this is a graph of the percentage of CPUs requested and actually used. So this is the request CPU numbers as a percentage more than are actually used. So 100% is where you would want this to sit at, or probably a bit above that for some wiggle room. But on average, we're using about 160% more CPU than we actually need. And on the memory side, it's even worse. We're using, on average, or get rid of the outliers because they're probably from runs where I've massively increased it for a specific reason, which I've forgotten. But get rid of those, and we're probably looking at 750% more memory than we actually need for, on average. Pretty horrific. So far, what we've done to combat this is to add a process tiny. There are umpteen jobs in the pipeline which are just simple bash scripts, so awk or find or very small stuff. These need next to nothing in order to run. We've got 100 meg there, but that's very much as you should never need more than this. Gene alignment has been modified to be slightly more aggressive than other tasks. We multiply the task attempt by 1.5, so it has a slight curve on its requirements. And many more will come as we collect more information from the summary stats. This is something that I've still got in fairly early development, really. But the important thing to note here is that we only found this because we were looking. There's obviously, I should imagine, all of us write Nextflow pipelines. How many of us are actually thinking about optimization of resources? How many further still are then collecting statistics on their resource usage? And then how many further are doing what we're doing and trying to generate something to improve that?
Yeah, there are solutions. Don't get too depressed. So Nextflow is highly configurable. So this is an example here where we can say, okay, so my default is going to be 30 minutes for each process, and then 1 hour for these ones, 2 hours for those ones, and so forth. So Nextflow allows targeting specific processes and giving them tuned resources, either in a constant manner like this, or you can make it depend on the input size. So this is an example where I say, okay, for smaller genomes here, I want 4 gigs of RAM, next category, 8 gigs, and so forth. We can make it a nice correlation against the input sometimes. So here, I want 50 megabytes of RAM for every 500 megs of sequence. This one, I increase the CPU, depending on the genome size and memory as well. So we can target specifically. I know something like this here, it means that, and I've checked that by looking at the source code, I can guarantee the usage of my tool is going to be within 50 megabytes of what I requested to the HPC. So that's very close to the optimum. In this case here, it's more about thinking about after multiple attempts, or thinking about the efficiency in long term of your processes. Multi-threaded jobs usually are becoming less optimal as you increase the number of threads. So you don't want to increase in linear manner, and instead I want to increase in a logarithm manner. That's what I'm trying to do here with BWMM. But then there could be a link with memory. Oops, sample sort. Where is it? Yeah, needs 768 megs of RAM per thread. So wherever I define here the number of CPUs, I need to add that here as memory as well. We need to think about how those things are linked together. But what we realize as well is that it's not just about number of memories the amount of resources you request for each process. It's about how the sub-workflow, the pipeline works. The standard NF core way is good. You've got one module doing one task, and then you run, so it runs one, makes an output file. That's then used for the second step, it produces an output file, and so forth. This is a very standard SAMtools sub-workflow that's advertised on the SAMtools documentation page. That bash pipeline cannot be shared through a core, so it has to live as a local module within a pipeline. So it's something that you need to be aware of. That's an issue, and then you need to do an extra step to go and implement it in your pipeline. But it makes massive differences because this is 5 times faster than having data separate jobs. And then you save 3 independent BAM files. So you save a lot of space as well. I've got another example here that's about parallelizing your BDOMM alignment across multiple machines. So there's a trade-off between modularity and performance. So we start with Netcore for modularity, and then as we optimize pipelines, probably we're losing a bit of that, but there are other benefits, obviously. So here, this is the outcome on 2 of our pipelines to do make statistics and figures for our publications called genome nodes and read mapping. You can see a total amount of memory requested across my test data set is divided by about 4. So we're using 4 times less memory as initially. Number of CPU reduces as well. So it's really the massive gains to be made. And like Damon said, if you overestimate your resources, it makes the whole HPC, the whole cluster go slower because you're taking resources away from other users. But I still have some questions there, and I hope we can discuss that. So for instance, so typically what we do is that when a process crashes, it will then restart with more memory, more CPU. But I would like to have to fine-tune that. If it's killed because it's using too much memory, then yes, I want to increase the memory, but don't necessarily want to increase the CPU as well. Or if it's killed because it increases the runtime limit, then I want to give it more runtime or more CPU, but not necessarily more RAM. Some tools may exceed because of internal error, like a segmentation fault. In that case, maybe I don't want to increase resources. It depends. I would like to have more flexibility there. What we're aiming so far is about finding resources so that every process works at the first attempt. So that would still be overestimating resources in some cases. Maybe it would be fine to actually decrease that amount and allow a few processes to restart. It's about, again, another trade-off there. I don't know how, we didn't do a raise your hand, so I don't know exactly how many of you are optimizing your pipelines, but we've got some outputs there. Can we share that with the community with NFCore? Is there space somewhere to deposit that? And then finally, we're doing this after having run the pipeline in an unoptimal, very suboptimal manner for several months now. Can we do that without running it at all? Is there a magic AI tool that can take a source code and tell me, how much memory it's going to use? I don't know. But I would like to avoid wasting resources in order to do the whole thing.
I will try to be very quick because I'm very aware that I've spoken far too much already. So as we've already said, CO2 footprint plugin, it's going to be very important for us. We need to be more sustainable anyway, because we're killing the planet, and it kind of gets the antithesis of DTOL. I need to generalize the script that I have so far. So at the minute, the script I have is very geared towards TreeVal. However, TreeVal it's a big pipeline that, anyway. A lot of graphs. We need more graphs. Efficiency per process. We need to be able to search through our data sets via date, pipeline versions, most pollutant, and the clades. And what should we do next? Should I turn this into a plugin so that it collects data on declared channels, and then it stores that data somewhere in a way that is easily that we can easily stick together and create massive data frames from? And is it possible to eventually add custom scripts pages to platforms? So then platforms is already there aggregating everything. So if we could do this, then we could have my script hook into it. Bob's your uncle. And finally, thank you.